Hi class, so today we are going to talk about skeletal muscles and electromyography, and we will also learn about some stretch reflexes. Now, since we are not going to do the actual lab experiments, there are some YouTube videos that I have posted along with a PowerPoint. Make sure that you go to those YouTube links and follow the experiments that other people have done and try to analyze some of the data that I have posted, and also think about the outcomes of the experiments. Okay, so we will start with a basic review of the skeletal muscle anatomy. So we know that muscle attached to bones. Here we see the biceps brachii attached to the humerus. Here we see the biceps brachii, which inserts into the radius. Now, if we think of a transverse section or a cross section of a muscle, we see that each muscle is made up of these bundles, as you can see, and each of these bundles is called a fascicle. If you pull out a fascicle, you will see that within each fascicle there are muscle fibers. So this is a muscle fiber and all these are muscle fibers. Recall that a muscle fiber is nothing but a fancy term for a muscle cell. Now if you zoom into each muscle fiber, as we do here, we see that each muscle fiber is made up of these structures, and these are called myofibrils. So here we see two myofibrils. Okay, and then if you zoom into myofibrils, you will see that they are made up of myofilaments, and there are two types of myofilaments, actin and myosin. On the diagram, on the right hand side, we do see a muscle which is made up of these units called fascicles. When we zoom into a fascicle right here, we see muscle fibers, and each muscle fiber is made up of myofibrils. We don't see myofilaments here, but we will look at myofilaments in our next slide. So to review, muscles made up of fascicles. Fascicles have muscle fibers. Muscle fibers are made up of hundreds to thousands of myofibrils. Myofibrils are made up of myofilaments. Two types of myofilaments, actin and myosin. I want you to know that actin is the thin filament and myosin protein makes up the thick filament. So here, this is one single muscle fiber, as you can see, and the membrane of the muscle fiber is called sarcolemma. This is the membrane of the muscle fiber. Now within the muscle fiber, these cylindrical structures, these are myofibrils. So this one is a myofibril, and each myofibril is made up of myofilaments, as we see the yellow and the brown structures. These are myofilaments. We do see a brown colored thin filament which is made up of actin, and then a thick filament in yellow here which is made up of myosin protein. Okay, all right. So as we talked about this before, we have myofibrils made up of myofilaments, two types of myofilaments, thick and thin. Hope you understand this. And also I have posted a link to YouTube video. This is a really good video that explains these concepts it explains these concepts really clearly. Okay, so we are zooming into the actin and the myosin proteins here, or we can say we are looking at the thin and the thick filaments here. So as you see, the yellow molecules, these are myosin. So this is a myosin molecule, this is a myosin molecule, And these myosin molecules, or the myosin proteins, make up the thick filaments. So we have the thick filaments as shown here, and then the red proteins are called actin proteins, and they make up the thin filament. There are other proteins associated with the thin filaments as well. As we can see, there is a green colored protein and a blue colored protein. The green protein that we see is called tropomyosin. You don't have to remember tropomyosin for lab testing purpose, but good to know for lecture. And the blue one is troponin. Troponin is the one that you must know because this is where calcium comes and binds. Okay, so to review, we have the thick filaments in yellow here and the thin filaments in red. The thick filaments are made up of myosin and the thin filaments are made up of actin protein. Good? All right, moving on. All right, so the thick and thin filaments are organized into specific units, and the units are called sarcomere. Know that sarcomere is the ultimate contractile unit of skeletal muscle. These are the contractile units of skeletal muscles. And if you look at a sarcomere, it extends from one Z disc, which is here, through another Z disc. So from one Z disc to the next, we have one sarcomere. If you look into a sarcomere, we do see the thick and thin filaments organized. Do you see that? They're organized in a definite pattern. And associated with the thick filament, do you see the green protein here? This is called titin or titin. It helps the thick filaments to be anchored in place. That's the function of titin protein. Okay, this diagram also talks about the A band, I band, M line. At this point, I don't want you to focus on these terminologies because you probably have covered this in anatomy, and we are not going to spend time on this anymore. Okay, moving on to the concept of motor unit. Now, a motor unit consists of a single motor neuron and the muscle fibers it controls. So it's not a motor neuron and muscles. It's between a motor neuron and muscle fibers. I want you to know that. So let's say, for example, let's say I am a motor neuron, and in lab, I am controlling 32 of you, right? So me, I'm a motor neuron, I'm controlling 32 of you. And this forms a motor unit. In this diagram, you do see a motor neuron, which is here. And the motor neuron is controlling 3 muscle fibers. Do you see? This is 1, 2, 3. So these 3 muscle fibers are controlled by a motor unit. I want you to understand that a motor unit can be small or big. If you look at a small motor unit, it'll be like 1 motor neuron controlling 5 muscle fibers. A bigger motor unit will be made up of, let's say, 1 motor neuron and 20 muscle fibers. In the example of me controlling the class, if I'm controlling 32, that will be a smaller motor unit. And if you think of me in your lecture room, me controlling 64 of you, that's going to be a larger motor unit. Good? Okay. Know that a motor unit works by all or none principle, meaning once a motor unit is activated, it will lead to contraction of the muscle fibers. Okay. And there is, so depending on what type of motion we are doing, there is fine control versus force. For example, let's say we look at a muscle, and a muscle is made up of 10 motor units. Now, if I'm going to pick up a paper which is super lightweight, I will actually activate only few motor units. However, if I want to generate a larger force, for example, if I am picking up a suitcase at an airport, I need a lot of force, so I will activate a lot of the motor units. Okay, if you have any further question on this, feel free to email me or participate in the class discussion board. Okay. All right. Moving on to the concept of neuromuscular junction. So let's say this is a motor neuron. Modern art here. So this is a motor neuron. And this is a muscle fiber that it controls. This is a muscle fiber. The junction between the motor neuron and the muscle fiber is called neuromuscular junction, popularly known as NMJ. So it's a region where a motor neuron comes into very close proximity to a muscle fiber. I want you to understand that the motor neuron is not touching the muscle fiber. It's in very, very close proximity. Okay. So what happens here? How does a motor neuron sends a command to a muscle fiber? This is what we are going to look at, and we will see what changes take place in the neuromuscular junction. All right, so I'll just write out here. So this is a motor neuron, this is a muscle fiber, and if I blow this up, this is what we see. This structure right here is the axon terminal of the motor neuron in yellow, and the red big structure here is the muscle fiber. The membrane of the muscle fiber, if you recall, is called sarcolemma. Okay, and we know from anatomy that the sarcolemma invaginates at specific locations to form these invaginations called T-tubules. Okay, so here a motor command comes down the axon, and the motor command reaches the axon terminal. When this happens, there are voltage gated calcium channels that open up, and when voltage gated calcium channels open up, calcium enters the cell. So we have an influx of calcium into the cell. Calcium causes these vesicles called synaptic vesicles to fuse with the presynaptic membrane, and exocytose or spit out acetylcholine into the space called neuromuscular junction, also known as synaptic cleft. So now we have acetylcholine floating into the synaptic cleft. Now what does acetylcholine do? Acetylcholine binds to specific receptors, which are shown in blue here. So acetylcholine binds to these receptors, and eventually these receptors open up their gates, and sodium from the extracellular space rushes into the rushes into the muscle fiber. Sodium, obviously, so acetylcholine binds to the ligand, gated sodium channels, and on the sarcolemma, and sodium enters the muscle fiber. Now sodium brings in positive charge and causes the fiber to depolarize, and if the depolarization is strong enough, it triggers an action potential that sweeps across the sarcolemma down the T-tubules. I will stop this video here and I will continue another video. Okay.